My name is Brianna Ames, and I'm a junior at the University of Delaware. I'm an animal science and agriculture and natural resources double major with a medical diagnostics minor, and this summer I'm one of the 2 emergency poultry disease interns.
The purpose of my project is to evaluate the efficacy of an open source and portable vehicle wash system to reduce the viral load of a contaminated vehicle. I picked up where a previous research intern left off by testing only foam-applied disinfectants to determine the best way to use foam with this system. An important part of farm biosecurity is to make sure that disease is not transferred from farm to farm. Our system is designed to be affordable at less than $500 and portable to make it easy to set up and take down to make it as accessible as possible to farms. In the trials, we placed metal squares that contain dried Newcastle disease virus, called coupons, on magnets in various locations on the truck. The vehicle wash system consists of a reservoir that contains water, foam concentrate, and disinfectant. When the system is operated, the mixed solution is pumped through the system, creating foam that is sprayed onto the vehicle as the vehicle is driven through the system, kind of like driving through a car wash. After waiting a 10-minute contact time to allow the disinfectant to properly act on the virus, the metal coupons are removed from the truck and placed in 6-well plates that contain a solution that stops the reaction between the disinfectant and the virus. Then the coupons are transported to the lab for further processing. In the laboratory, the coupons are scraped and agitated to dislodge any remaining virus from the coupons, and the solution from each well is pooled, diluted, and injected into embryonated eggs. The eggs are incubated for 5 days, and then they are cold shocked to euthanize the embryos, and fluid is drawn to be tested for the presence of live virus.
If the virus survived the exposure to the foam disinfectant, the virus would replicate in the eggs. To determine if virus replication occurred, red blood cells were added to the fluid to see if virus particles attached to the red blood cells, causing agglutination. Agglutination causes a well to become cloudy due to the clumping of red blood cells caused by the virus attaching to them. This is called the hemagglutination test. If the red blood cells agglutinate together, that means that the egg does have live virus present. To confirm the agglutination was caused by Newcastle disease virus, an additional test is performed called the hemagglutination inhibition test to confirm that Newcastle disease virus caused the clumping and not something else. This involves adding Newcastle disease virus antiserum to the egg fluid before adding the red blood cell solution. This allows the antiserum to bind to the virus and prevent the now antibody-coated virus from being able to attach to the red blood cells and cause agglutination. If the red blood cells are not bound by the virus, they will settle at the bottom, and this signifies the clumping agent is the Newcastle disease virus.
Going forward, we are using Emma Redmond's evaluation research of the undercarriage wash system to create more effective solutions to run through the system. We are currently testing various types of disinfectants, including phenolics and oxidizers, along with different concentrations of foam.